Hello everyone, today we will discuss Plasmodium vivax life cycle. Plasmodium vivax life cycle. So the Plasmodium vivax is a digenetic parasite. Plasmodium vivax is a digenetic parasite. Digenetic parasite means they contain two hosts. One host man, another host mosquito. Female Anopheles mosquito. They contain a two host. One host man, another host female Anopheles mosquito. First one, so the life cycle in man. Plasmodium vivax life cycle in man. So in the Plasmodium vivax, reproduce in man, reproduce asexually. Asexual reproduction. So the asexual reproduction is called as schizogony. So the Plasmodium vivax in man reproduce asexual reproduction. So this asexual reproduction is called as schizogony. The asexual reproduction is called as schizogony. Schizogony means asexual reproduction. So the Plasmodium vivax in man reproduce asexual reproduction. So the asexual reproduction is called as schizogony. So in man they are living in these schizogony in the two cells. One cell liver cells and RBC, red blood cells, liver cells and red blood cells. So the liver cells is called as hepatic cells. Liver cells is called as hepatic cells. So these hepatic cells, in the hepatic cells, the reproduction is called as hepatic schizogony. This reproduction is called the hepatic schizogony. So the in liver cells the reproduction is called as hepatic schizogony. So the next one RBC. So red blood cells. In the red blood cells the reproduction is called as erythrocytic schizogony. Erythrocytic schizogony. So these are Plasmodium vivax, the intracellular, they are living in two cells. One is the liver cells, another one is the red blood cells. So the liver cells, the asexual reproduction. Is called as hepatic schizogony. So in the RBC, asexual reproduction is called as erythrocytic schizogony. Hepatic schizogony and erythrocytic schizogony. Two sided things. One is the hepatic schizogony. Second one is the erythrocytic schizogony. Hepatic schizogony occurring the liver cells. So erythrocytic schizogony occurring the red blood cells. First one hepatic schizogony. Hepatic schizogony means the asexual reproduction occurring in the liver cells. So they are called as hepatic schizogony. So this hepatic schizogony is proposed by Short and Gurnham. So the hepatic schizogony is proposed by the two scientists. One is a Short, another one is a Gurnham. These two scientists proposed by the hepatic schizogony. Short and Gurnham proposed by the hepatic schizogony. So first step, the female Anopheles mosquito A female Anopheles mosquito, a female Anopheles mosquito infected Plasmodium vivax. Plasmodium vivax. So the a female Anopheles mosquito infect the Plasmodium vivax, infecting the Plasmodium vivax. So the female Anopheles mosquito infected the Plasmodium vivax. So these infected female Anopheles mosquito bites a man. Infected mosquito bites a man. The infected mosquito bites a man. So these mosquito saliva they contain sporozoites. Mosquito saliva contain sporozoites. The mosquito saliva contain sporozoites. These sporozoites enter into the human blood circulation. Sporozoites enter to the human blood circulation. So they are entering to the human blood circulation. Once revised, so the infected female Anopheles mosquito, a female Anopheles mosquito affected by Plasmodium vivax, affected by Plasmodium vivax. So these mosquito bites a human being. So these mosquito saliva contain sporozoites. That sporozoites entering into the human blood circulation. So the one mosquito bites a human beings. The sporozoites, 2000 sporozoites entering to the human blood circulation. 2000 sporozoites entering to the human blood circulation. So within half an hour, within half an hour, the sporozoites reach liver cells. So the mosquito bites a man, that saliva contains sporozoites. That sporozoites, 2000 sporozoites entering into the human blood circulation. Within half an hour, these are sporozoites entering into the liver cells. Within half an hour, these sporozoites entering into the liver cells. Entering into the liver cells. Next one, so in the liver cell, these are the two types of the schizogony. One is the pre-erythrocytic cycle. Pre-erythrocytic cycle. Second one, exo-erythrocytic cycle. Exo-erythrocytic cycle, pre-erythrocytic cycle and exo-erythrocytic cycle. Pre-erythrocytic cycle and exo-erythrocytic cycle. So in the liver cells, there are the two types of the cycles. One is the pre-erythrocytic cycle, another one is the exo-erythrocytic cycle. Pre-erythrocytic cycle and exo-erythrocytic cycle. First studying pre-erythrocytic cycle. First one, pre-erythrocytic cycle. Pre-erythrocytic cycle. Pre-erythrocytic cycle. First one, pre-erythrocytic cycle. So pre-erythrocytic cycle means, so the first step, the sporocyte entering into the liver cell. The sporocyte entering into the liver cell. So these are the liver cell. These are the sporocyte. The sporocyte entering into the liver cells. Next one, these sporocytes that converting to the tropocyte. They are converting to the tropocyte. So first step, the sporocyte entering into the liver cell. Next one, they converting to the tropocyte stage. Next one, the tropocyte stage, they eating to the cell organelles. They are the liver cell. These are the liver cells. So in the liver cells contain a liver organelles, cell organelles. So these tropocyte stage eating to the cell organelles. Next one, they are assume the increasing in size. So they are the increase in size, increase in the body size, increase in the body size. So this stage is called a Seijan stage. This stage is called as Seijan stage. Next one, these Seijan stage, the nucleus and cytoplasm. So these are the cytoplasm. So there are the many mitotic divisions. So this tropocyte stage. So this stage, many mitotic divisions is called as schizogony. Schizogony means cell division. They are divided into many cells. So this stage is called as schizogony. Schizogony means asexual reproduction. Schizogony. Finally, the final step. So the liver cell is bursting. Releasing 12,000 cryptocytes. Finally, the liver cell is bursting, releasing 12,000 cryptocytes. So these are called as cryptozoites. How many number? 12,000 cryptocytes. 12,000 cryptocytes releasing from the bursting liver cells. So these are called as pre-erythrocytic cycle. Once revised, first step, the sporocyte entering into the liver cell. Next one, these are transforming into the tropocyte stage. They are changed into the tropocyte stage. The tropocyte stage eating to the cell organelles. They are assuming increasing in size. This stage is called as Seijan stage. So this Seijan stage, the nucleus and cytoplasm, many mitotic divisions. So they are the division is called as schizogony. Finally, the liver cell is bursting. Releasing 12,000 cryptocytes. So these cryptocytes is also called as first generation merocytes. The cryptocytes is also called as first generation merocytes. So these are called as pre-erythrocytic cycle. So this cycle is called as pre-erythrocytic cycle. Pre-erythrocytic cycle. Pre-erythrocytic cycle. So these pre-erythrocytic cycle, the time period 8 days. Time period 8 days. This time period 8 days. Next one, these cryptozoites, the two options. These cryptozoites, two options. One option, these are entering into the RBC. These cryptozoites entering into the RBC. So these are the RBC. The cryptozoites entering into the RBC. So they are continuing to the erythrocytic cycle. So this cycle is erythrocytic cycle. Next one. So second option. Another option. These cryptozoites entering into the new liver cell. These cryptozoites entering into the new liver cell. Entering into the new liver cell. Second option. These cryptozoites entering into the new liver cell. Entering into the new liver cell. They continue to the exo-erythrocytic cycle. Second one, second heading, exo-erythrocytic cycle. Second one, exo-erythrocytic cycle. Exo-erythrocytic cycle. Second one, exo-erythrocytic cycle. First one, pre-erythrocytic cycle. Pre-erythrocytic cycle, the first step, sporocyte entering into the liver cell, converting to the tropozoite stage, increasing size, Shoijant stage. Next one, seizogony. Finally, liver cell bursting, releasing 12,000 cryptozoites. So these are called as pre-erythrocytic cycle. This entire process completed in 8 days. So these cryptozoites, two options. One option entering into the RBC, they continue to the erythrocytic cycle. Continue to the erythrocytic cycle. So the second option, these sporozoite entering into the new liver cell, continue to the exo-erythrocytic cycle. Continue to the exo-erythrocytic cycle. So these cryptozoite entering into the new liver cell. Next one, these are converting to the tropozoite stage. So they are converting to the tropozoite stage. Tropozoite stage. They are converted to the tropozoite stage. Next one, these tropozoite stage eating to the cell organelles. Eating to the cell organelles. These are increasing in size. Increase in the size. Increase in the size. So these are called as Shoijant stage. Shoijant stage. Increasing in size. So they are called as Shoijant stage. Next one, these Shoijant stage, the nucleus and cytoplasm, the many mitotic divisions. Many mitotic divisions. So the after many mitotic divisions. Many mitotic divisions. So they are increasing in numbers. Many mitotic divisions. So they are called as seizogony. Seizogony. Seizogony means asexual reproduction. Simple. First one, the cryptozoite entering into the liver cell, they transform to the tropozoite stage. The tropozoite stage eating to the cell organelles, they increasing in the size. So they are called as Shoijant stage. The Shoijant stage, many mitotic divisions. So they are increasing in the number. So they are called as seizogony. Seizogony. Next one, the final stage, the liver cell is bursting. The liver cell is bursting, releasing two types of the cryptozoites. Releasing two types of the cryptozoites. One is the large cryptozoites. Large cryptozoites. Another one is the small cryptozoites. Large size and small size. So they are the large and is called as macro metacryptozoites. Large size is called as macro metacryptozoites. Macro metacryptozoites. Macro metacryptozoites. Next one, small size. The small size cryptozoites is called as micro metacryptozoites. So these are called as micro metacryptozoites. Metacryptozoites. So the final step, first step, cryptozoite entering into the liver cell, transfer to the tropozoite stage. Next one, Zygote stage. Next one, Zygote. The finally, the liver cell is bursting. Two types of the merozoites is releasing. Two types of the merozoites is releasing. So the large size merozoites is called as macrometacryptozoites. Macrometacryptozoites. The small size is called as micrometacryptozoites. Macrometacryptozoites and micrometacryptozoites. So these are called as exo-erythrocytic cycle. Exo-erythrocytic. Exo-erythrocytic cycle. So this exo-erythrocytic cycle is complete in 2 days. This is complete in 2 days. Exo-erythrocytic cycle complete in 2 days. Next one. So these micrometacryptozoites. The micrometacryptozoites, these are entering into the new liver cell. They are attacking into the new liver cell. The macrometacryptozoites entering into the new liver cell, they continue to the exo-erythrocytic cycle. The macrometacryptozoites entering into the new liver cell, continue to the exo-erythrocytic cycle. Next one, the micrometacryptozoites. So they are the micrometacryptozoites. The micrometacryptozoites entering into the RBC. The micrometacryptozoites entering into the RBC. So they are continue to the erythrocytic cycle. Continue to the erythrocytic cycle. So they are the pre-erythrocytic cycle and exo-erythrocytic cycle. So the pre-erythrocytic cycle complete in 8 days. Exo-erythrocytic cycle complete in 2 days. So the pre-erythrocytic cycle final step releasing 12,000 cryptozoites. 12,000 cryptozoites. So they are contained two options. One option entering into the RBC, continue to the erythrocytic cycle. Second option, they are entering into the liver cell, continue to the exo-erythrocytic cycle. So in the exo-erythrocytic cycle, the final step releasing two types of the cryptozoites, macrometacryptozoites, micrometacryptozoites. So the macrometacryptozoites, they entering into the new liver cell, continue to the exo-erythrocytic cycle. So the second one, micrometacryptozoites. So they are entering into the RBC, continue to the erythrocytic cycle, continue to the erythrocytic cycle. So these are the hepatic schizogony. Hepatic schizogony. So these hepatic schizogony, there is no causing the symptoms, there is no affecting the host, but these function increasing of the numbering, increasing of the numbering. Next one, pre-patent period. Next one, studying pre-patent period. Pre-patent period. So 2 marks, very very important. Pre-patent period. The pre-patent period means interval between the first entry of Plasmodium in the blood. First entry of Plasmodium. First entry of Plasmodium in blood, the cryptozoites. First entry of Plasmodium in the blood of the form of cryptozoites, and second entry of Plasmodium. Second entry of Plasmodium in the form of cryptozoites. In the form of cryptozoites. This time period is called as pre-patent period. The time period is called as pre-patent period. First entry of sporozoite in the blood. So this sporozoite entering into the blood. So first entry of sporozoite in the blood to the second entry of sporozoite, second entry of Plasmodium in the blood in the cryptozoites. This time period is called as pre-patent period. Pre-patent period means first entry of that interval between the Plasmodium in the form of sporozoites entering into the blood, and second entry of Plasmodium in the blood in the form of cryptozoites. This time period is called as pre-patent period. The time period is called as pre-patent period. So this time period in the 8 days. This time period is 8 days. So they are the pre-patent period. Next class we will discuss erythrocytic cycle. Next class we will discuss erythrocytic cycle.